Yeah, I'm live on Facebook.
Okay, good morning. It's 11 o'clock, August 1, Sunday. The first Sunday of August, we should have been at Prithvi Theatre. Such a beautiful venue, but unfortunately, because of the pandemic, we are still virtual. But this session is really special. We have with us Professor Uma Ramakrishnan from the National Center of Biological Sciences, which is part of TIFR in Bangalore, where she works on wildlife biology, conservation, tigers, and lots of exciting things. And actually, I'm going to let her tell us exactly what she does. And over to you. So, because, there is so much that I don't understand really what's ecology, what's conservation, what's biology, what's, all these things. But let me tell you that Uma has, of course, been working in the field for almost 2 decades, following a variety of animals, interacting with a lot of different stakeholders, doing her research, of course, but also interacting with policymakers, government, etc. And in 2020, she got the Homi Bhabha Award for Science Education. She was one of the winners. So, she's also a fantastic communicator, and she's written a new book that she's going to tell us about, or being part of, a team that wrote a new book. And so it's over to you. And yeah.
Thanks,
Let's get started. Now, remember, only one thing before we start, Uma, is we can't see the audience. So we'd love it if you could tell us where you're watching from, and whether you're a school student, college student, just a professional. Just put it in the chat. And whenever you want to ask us questions, please put it in the chat if you're on YouTube or Facebook. If you're on Zoom, of course, you can use the Q&A box. And if you really want to ask a question at the end, we'll of course allow you to talk and ask a question. So I'll also put the Zoom links in the Facebook and YouTube in case people want to switch over to the Zoom. So yeah, with that, over to you, Uma. Let's get started.
Thanks, Arnab and the Chai and Wai team for having me here today. It's really fun. And as Arnab said, I do wish I was also at Prithvi Theatre. It's a really fabulous venue, and hopefully that will also happen relatively soon in the future. So I'm Uma, as Arnab said, and as he said, I've been spending quite a lot of time with my lab exploring different corners of India and trying to understand more about wildlife. And I'll present some things to you, but I wanted to start by saying that motivation for all of our work is really a fascination for nature. All of us feel from a very young age, we love animals, we feel a fascination for nature. We are soothed by green, we feel happy seeing trees, and even soothed by deserts and their beauty, their stark beauty, or the high altitude and its stark beauty. Ullas is in the high altitude right now, so I'm sure he appreciates that. And I think that, as scientists, we're supposed to be very, test hypotheses and, very kind of sounds very formal, right? But I think that the best part about being a scientist is actually just having fun and trying to find out things and understand things driven by your curiosity. So I guess that the work I'm going to present is also something which started with curiosity. So when I was going to start my job almost 15 years ago at NCBS, I just happened to read a paper about tigers. And the paper, which was looking at tigers across the world, had a small section on tigers in India. And, it said nothing very distinctive about Indian tigers. And I was really surprised because I thought, almost at that time, 50% of the world's tigers, this is 2005, 50% of the world's tigers are in India. And yet they seem quite unremarkable, not much of genetic variation. They don't seem to be saying much. And so that began the journey of how do we understand more about tigers? And of course, the first hurdle was how do we sample tigers? How do we get material from tigers? How do we study tigers? And so there began the journey of my tryst, in some sense, with fecal samples or poop. But if ever we get a chance to go together to the field, or if you have a cat or a dog, I'm sure you'll realize that it's actually very interesting to look at poop. Maybe not domestic cats and dogs because they eat cat food and dog food. But otherwise, poop is actually a treasure trove and provides us a lot of information. But I started off by saying that, what motivates a lot of us is this love for nature. And as we think about nature, it's almost an immediate thought. We realize that nature is really in peril today, more than it has ever been before. And so immediately then we have to not only think about understanding, but thinking about a future for nature. Okay, so I'll just start sharing now, and please interrupt me at any time, because it's much more fun to interact. I mean, it's very boring if I just keep speaking all the time, and if you don't interrupt, I'll just keep speaking because I speak a lot. Okay, so basically, before we get started, just a nice picture to inspire us. This is a tiger from Ranthambore. In the background, you can see these beautiful pink buildings. So Rajasthan, Ranthambore is in Rajasthan, about 3 hours from Jaipur by car. And, those are erstwhile hunting lodges. So there was a time when these were prime areas for hunting. And you can see it's kind of interesting to see the tiger in the foreground with a lake in the middle. And basically what's really interesting about tigers is they're actually huge. They are the largest, land carnivores that exist on Earth today. So males are about, anywhere between 2.5 to 4 meters, and females are, of course, smaller at about 2 to 3 meters. But a male can weigh up to 300 kgs and a female about 170 or so kgs. So just to get an idea of how big these animals actually are. And as I said, something which we all think about, and often for me, this is something which I think about very acutely. So many times I feel like I have this, feeling, a conscious feeling all the time that, biodiversity is at risk. And I feel like shaking everybody on the road and saying, don't you realize it? Shouldn't we do something about it? There was a question about this, and so we'll talk about that a little later. But, biodiversity, it's not just about pretty things like tigers. Things like tigers get a lot of attention. And so sometimes we can dismiss that, it's just about nature, but it's just about, some feel good stuff. But it's not like that. The water we drink, the air we breathe, the fruits we eat, everything comes to us from biodiversity through ecosystem services. And if we don't have that intact nature, if we don't have that biodiversity, we will lose these services. So it's a cost. You're getting it free right now. And if it's lost, it's a huge cost that we will pay. And the best example is the last 1.5 years with COVID-19. This is an example of a spillover, right, where there was, diseases in wildlife, which have then eventually somehow spilled over to people, and basically has caused tremendous, economic loss as well as social and, a lot of issues for all of us in the last year. So basically, intact biodiversity is really critical for human well-being. And it's something which we should all be cognizant of. We should all think about a lot. And it also so happens that, despite in general, you open the paper, you will see a lot of discussion about wildlife, or you'll see discussion about nature. But the reality is that 1 in 4 species of, say, mammals and 40% of amphibians are threatened with extinction today. And to me, this is something very, I think almost personal, because the International Union for Conservation of Nature and the associated law called CITES Convention for International Trade of Endangered Species, which bans hunting, which bans, sale of species and so on. The legal framework which allows the protection of biodiversity was launched in 1972, and that was the year I was born. So it's almost like, despite almost 50 years of very kind of a global interest in something like conserving nature, the reality is nature is declining, species are declining, and extinctions are we are seeing extinctions, and they are more likely to happen in all of your lifetimes as well. And in the context of tigers, this is a picture from the times of the British Raj. Tigers were hunted as sport, and also the British did classify them as beasts and vermin. And so you were given at one point, not recently, a bounty or a reward for hunting tigers. So this feeling, say, that I have, and maybe many of you have, that nature is important, we should value it, we should save it, is relatively recent in human history. And even up to 100 years ago, that was not the case. So I'm contradicting myself. I just said, the last 50 years, we're supposedly thinking about these things, but still biodiversity is declining. But it was even worse, 100 or so years ago. And you can imagine, there are some beautiful books on ecological history by Mahesh Rangarajan, for example, which talk about Palam, which is the site now of the Delhi airport, where tigers roamed, even less than 100 years ago. So tigers were really, when you say India, the land of the tiger, yeah, there were tigers really literally everywhere in India, even just 100 years ago. Also, there is ongoing trade of tigers. So tigers are used, tiger parts are used in traditional Chinese medicine. Tiger skins are, of course, beautiful, and so used for purses and so on and so forth. And so this illegal trade, I said illegal because, remember, I just mentioned the CITES, Convention of International Trade for Endangered Species, which makes it illegal, is ongoing. And in the end, I'll tell you a very funny story, which we were involved in, about a tiger cub in San Diego, and how we helped a little bit over there. So all of these pressures on tigers, or hunting in the past, ongoing hunting, though illegal, still ongoing hunting, ends up in basically their extinction. So if you look at this map of the tiger range, so we're not talking here about a very finicky species, a specialist which lives only in a specific type of rainforest, like say king cobra only lives in, very wet rainforests. No, tigers live from, tundra, like very cold environments in Siberia, to, tropical rainforests in Sumatra, to the dry deciduous forests of India, and even, mangroves like the Sundarbans. They also had a very big range in this part of, erstwhile Russia. But the Caspian tiger, for example, went extinct in the 1930s, the Bali tiger in the 1940s, the Javan tiger in the 1980s, and the South China tiger, huge range in South China in the 1990s. Now, these are not different species. It's all just one species. It's the tiger. These are just populations, but still, for a species which occupied a large area, it's now reduced to 95% of its range. Now think about this. If you are as humans, look at how cities grow. We keep expanding. We're always occupying more areas. Imagine if you were forced and pushed into a much smaller part of where you used to live, where your house used to be. And that's what's happening to a lot of biodiversity. Even, for example, with bats, we demonize bats today, but they're cutting trees, they lose their habitat, and so they end up sometimes coming in closer contact with humans in searching for new habitats and searching for areas to live. And so, as I said, the IUCN, on the International Union for Conservation of Nature, depicts, classifies tigers as endangered. And there are criteria to do this. This is not just like, oh, it's beautiful, and so let's just say there's not many, so let's just call it endangered. No, endangered species have to have declined by so much, have to have lost so much area, and so on. So there are different criteria. So when we think about conservation and classifying species at conservation risk, what their conservation risk is, it is quantitative. It is sometimes there's not very good data, but overall the criteria are quantitative as to why something is endangered or not. Okay. And we've been talking about extinction. I don't want to go into too much detail about these graphs, but I just want them to be there so that you can see what this is about. So talking about extinction, we all know the word, and then we think we should also understand why extinction happens. So why do populations or species go extinct? So when as a scientist, the first thing you have to do is you have to explore this question in a system which is not going extinct. You can't just study something which is going extinct and understand why extinction is happening, right? So what you do is you can study it in a very common species, like say this butterfly, in this paper, where they actually showed with many butterfly populations that the chance of extinction or the probability of extinction decreases as the population size, how many butterflies there are, increases. And there's a log scale, but you can see that, if you have population size of 100 or more, that's like sort of like a threshold. Generally, extinction probability is pretty low, right? So if you have small populations, the chance of extinction are high. Why this is, if you're interested, we can talk about later, but this is an observation. This is a pattern that when populations are small, the chances of extinction are higher. And like I said, maybe we can just remember this number 100, okay? On the other hand, when populations are isolated, the chances of extinction are higher. This is again an experiment with butterflies. And don't focus too much on the graphs, but basically you can see on the X here, or what you're changing in the experiment is the number of patches, number of populations, but also here you have many patches which are not connected to each other. And here you have patches which are connected to each other. Now, we said small populations are likely to go extinct, but if you have lots of small populations, right, which are not connected to each other, then also extinction probability is high. So when you have lots of fragmented populations, lots of different populations which are not talking to each other, the chances of extinction are high. And the last thing which affects extinction is something that I work on, which is genetic variation. You often must be hearing statements like, oh, genetic variation is like money in the bank, right? Why? Why is it like money in the bank? Why is it important to have genetic variation? Well, when the environment changes, which it constantly does, if you have lots of variation, then something in that variation is useful to counteract the new changes in the environment. So it's just hanging around that variation. But if it's not there and you're only of a certain kind, then as the environment changes, you're not able to adapt and your chances of extinction are high. Okay, so this again is shown with data on butterflies, that when you have high heterozygous loci, that just means more variation. You can read this as low variation and high variation. When you have low variation, the chances of extinction are high. When you have high variation, the chances of extinction are low. And the final thing, which is basically an interaction between these, is that when you have very small populations, you also have inbreeding or mating between relatives, and this also leads to extinction. So it's what's called an extinction vortex. When you have small isolated populations, they are hit by so many things, including inbreeding, low genetic variation, some chance events, environmental change, etc., that they're just funneled, they're like pushed towards extinction, right? So this is kind of a generic conceptual understanding, right? But then what we need to know is how small are populations, how connected are populations, right? We need to know and if they are small, what is making them small? What is stopping them from growing and becoming larger? What is making them disconnected, and so on. So this is a general framework which helps us understand extinction for any species. But if you're talking about tigers, which I started off with and we'll discuss, then what is it that drives tigers into this realm of extinction? So now let's look at India. This is a map of India and broadly tiger range in 1700. The green is, kind of green cover, it could be forests, etc. This is 1800, 1900, and 2000. So imagine, it's not strictly true, but imagine the tigers were tied to the green. Of course, just their habitat has decreased, the amount of area, they have become more isolated. See, this is so much more continuous, right? The green patches are flowing into each other, but this is completely disconnected. There's very little, green which is connected. Okay. So basically what we need to know, hopefully to prevent extinction, it's a very big thing, probably not possible. But what we need to know is where tigers are, how many there are. We need to ensure that these populations are not too isolated. And this is almost like, very police stuff. We need to understand, we need to make sure that they are protected. And I'll show a little bit about that towards the end. So how do we usually count tigers? I do genetics, but there's a lot of other ways to count tigers. You read about it in the paper all the time. India has 3,000 tigers, 4,000 tigers, etc. Where do these numbers come from, right? That's the first thing we have to ask. So luckily for us, this is a very cute picture of a cub. And in the background, you can see a camera trap. Okay. So what they do in the forest, they put these camera traps. And when the tiger walks through it, there's an infrared beam which is broken. And so the cameras take pictures on both sides of the tigers. And the person who pioneered this method to count tigers is Ullas Karant. He's a famous tiger biologist. And he, over many, many years, working in the Western Ghats, collected many, many pictures of tigers like this. And initially, they would compare tiger pictures and say, this, of course, tigers have unique striping pattern, right? So they would compare pictures and say, same individual, different individual, same individual, different individual. And then when you have enough camera traps across the forest, you have what is called mark recapture. That is, you have a capture, you saw that tiger, and then somewhere else, maybe a little bit later, another recapture of the same individual. And given that you're sampling with these camera traps, you've put them in specific locations, you can use a statistical framework to say, how many times have I caught and re-caught the same individual? I can use that to estimate how many tigers there are in this park. Simple example, suppose you're catching mice, you catch them, you catch 5 mice, you paint their nails and you let them go. And you catch 5 mice again, and 1 mouse you caught had painted nails. So that means there's 20 mice. Okay, just ratio and proportion, very simple, but there's much more complex models for this. But the idea is like that, right? So the first thing that we did, and of course, I should not, now this whole exercise has been taken up by the government of India. And recently, the government of India was in the Guinness Book of World Records for the most massive conservation effort in the world to count tigers using camera traps across the country. So it's like a huge effort with camera traps all over and people, walking and collecting all this data. So just as, this tiger has unique stripes. So why does a tiger have unique stripes? Well, it has something to do with its genetic material. We don't know exactly, but, we all know this, that we all have DNA. Every cell of our body has a nucleus, and in that nucleus is stuffed, packed a whole lot of DNA, right? And basically, this DNA helps us to answer questions like, where are tigers? Why? Because tiger DNA is different from leopard DNA. So if I can design my experiment to distinguish tigers from leopards, if I get some DNA from the field, I can say, oops, it's a tiger. No, it's a leopard. No, it's a dhole or a wild dog, and so on. How many are there? Every tiger has a unique DNA sequence, right? And if I look at the whole, of course, the whole genome is like a big book. I can't always read the whole book. That's too much. But I can focus in on specific words in specific chapters and compare those words across the different samples. If that string of words is exactly the same, then I can say, oh, the same individual. If it's different, it's a different individual. I can also assess whether tiger populations are mixing. Why? If there's two populations which are not mixing at all, their DNA signatures will be very different. But if they are mixing, they'll be similar. And using these kinds of tools, I can also try to understand how they can be protected. Okay, so basically what we do, we go into these forests, and luckily for us, I hope you've been to protected areas in India. They are spectacular. And luckily for us, tigers, they walk on the forest roads. Okay. So, they don't like live on trees and all that stuff. Would be very tough. Studying monkeys, like imagine trying to collect fecal samples of monkeys. You're waiting, you're standing under the tree. Okay. It's not easy. And some other cats and small cats, they generally are, much more off the road. Tigers, they walk on the roads inside the forest. If you've ever been to a protected area, you might have gone on a safari, and the safari vehicles also take the same roads. So basically, they're mud roads, but they are, all of our protected areas have a network of roads through them. And the tigers also happily walk on these roads. And as they walk on the roads, they mark their territory in two ways. One way is they spray. So they spray urine. And one of the projects we're doing now is we're trying to understand the chemical composition of that urine, because long ago there was a researcher called Brahmachari who said, oh, this urine smells like basmati rice. And he isolated some compounds which he suggested was the reason it smells like basmati rice. And today, people use, in management, they use obsession for some unknown reason to try and attract tigers. So we have no idea what this tiger urine really has, what attracts tigers, is an attractant, repellent, and so on. So anyway, so that was an aside, but we're trying to even understand something like that. But as we walk through these forests, we may not see tigers. We may not see tigers. That's something which, sometimes with students, they think that working on tigers means we'll be seeing a lot of tigers. Not necessarily, some places, but not everywhere. But we do see a lot of scat or fecal samples of tigers. And we collect the scat. We don't collect the whole thing. Why? Because the cells of the tiger, where the DNA is, I hope I didn't make that jump very quickly, but we want to access the DNA of the tiger, right? Now, to get the DNA of the tiger, we need like tiger cells. Even if they're broken, it's okay. DNA is very stable. I'm sure you've read about these ancient DNA studies of mammoths and Neanderthals and so on. It survives for thousands and thousands of years, right? That's the thing. That's a challenge with the chemical sampling of urine, because chemicals, often some of them are very volatile. You can't capture them for a long time. But these samples, they basically, as the fecal sample passes through the intestine of the tiger, it pulls off cells. And those are tiger epithelial cells from the tiger's intestine. Those cells have tiger DNA. So when you see a fresh scat, you will see the top part is kind of a little shiny with mucus. And that is where the tiger cells are. If I break the scat and start pulling out stuff from inside, that is where I'll find DNA of prey. And in fact, we do that when we're trying to understand tiger diet. For example, we're trying to understand whether in some protected areas, tigers depend more on cattle than on wild prey, right? Then we would go inside and take the inside part. But since we're trying to look at tigers, we tend to we just take like a Johnson's earbud, but of course a sterile swab, and we just roll it on the scat, put it into a vial, and bring it back to the lab. And really, as Arnab sent a picture earlier, my lab is stuffed with scats. And, you know, a joke in my institute, because, you know, my institute, you know, is Tata Institute of Fundamental Research, and a lot of people doing very, very amazing science, but certainly their labs are not filled with poop, right? And so the common joke is, oh, Uma and her poop lab filled with scat. But really, these are, you know, these samples are really treasure troves of information. So let's go back to what India is today. Arnab, you want me to stop in the middle and answer questions?
It's entirely up to you. Actually, as of now, I do think there's just one comment. There's just one comment which has come many times, the same comment. But I did have a question for you. I mean, you said that when you collect these samples, if you collect it from the outer side, it's mostly some cells which have been shed along from the epithelial part. Will these decay? I mean, do you really need fresh scat to look at it?
They do decay. It does decay over time. And we've tried, for example, to understand, you know, what conditions, how long and all. But it's difficult to see. It depends a lot on the environmental conditions also, how much decay happens. When it's very, if it's hot, it's okay. It doesn't affect the degradation or the decay of DNA so much. But if it's wet, that is bad. So usually in the rainy season, or when it is in areas which are very wet, we have very low success. So in fact, recently we were working with a team in Indonesia, and some of the pictures of tiger scat they sent me were green, like they have so much of fungus and stuff on them, and they've just collected them. I don't know, for example, those kind of conditions, what success we'll get. We just recently started some work on snow leopards. We are helping the government of India count snow leopards in Jammu and Kashmir. And there's a lot of UV at the high altitude, which definitely does degrade DNA, right? So it could be that we get very poor results there. We don't know. The thing is that there's a lot of variation in every environmental condition. So setting up an experiment in the lab, I'm not sure how well it would work unless we put rain on the scat or, you know, something like that. But it does vary a lot. It does vary how successful we are.
And how much do you need to be able to identify? You know, do you take like 1 spoon, 2 spoons, just to give people an idea of how much you actually collect?
Yeah. So basically, we now
how much poop does a tiger make?
Lots. So usually,
we've seen cows or something. We know how much cow, typically.
Yeah, definitely herbivores produce much more poop. Like elephants, oh my God, they poop so much. But if you look at carnivores, they don't poop as much. And usually they eat once, tigers eat once in 5 days or so, something like that, roughly. And after they eat, their first excreta is usually very liquid. And then all the bones and the hooves and all the big stuff starts coming out. So then they would probably poop once a day or something like that. So, and then they'll eat again. So that's how it usually is. And tigers,
by looking at the inside, you can also say if it ate a deer or whatever, goat or something else.
Yes. Oh. So that's what we're doing, for example, to understand things like conflict, where there's a lot of conflict. There are already populations. For example, if you look at the lions in Gir, they are subsisting on livestock more than wild prey. So a lot of questions then come, like, are these animals wild if they are just eating cows? I don't know the answers to those questions. It's something we have to think about. And also remember, these cows are somebody's cows, no? If they're eating cows, they are killing a cow which belongs to a farmer or something. Of course, there are compensation schemes, but people who live outside protected areas face a lot of these challenges, which we cannot discount. So yeah, I hope that answers your question, Arnab. We usually collect just, a few. We don't collect scat anymore. We just rub the surface with this, these swabs, put it into a small vial and shake it And then from that, it's basically a buffer or a solution which has SDS, which is a soap, and breaks cells, right? So then we just, from that, we have 1 ml, and from that 1 ml, we can identify individuals, identify species, and whatever we need to do. So it's not that much material, if that's sometimes it doesn't work because maybe the scat is very old, or, for example, if we see fungus, or if we see it burnt, because sometimes there's forest fires, if we see it with beetle activity inside the scat, beetles roll up the scats, then we know it's definitely not, if it's definitely older than 2 or 3 days. Very fresh, so we can tell. Yeah.
Let me just quickly look at YouTube. What is scat? Somebody has written already. Scat is the fecal matter of tigers, basically poop, shit, whatever you want to call it. Okay, good question from Priyatosh asking, does the texture of the poop tell you an idea of the health of the tiger? Or will you come to this later?
Yeah, no, I can answer that. I don't think so. I don't basically, like I said, the first excreta is always liquid after they eat. So you could think that they may have some digestive issues by looking at just that, but it's not the case. But we don't know probably enough about individuals, like we don't follow individuals and their pooping routines to really probably say that very scientifically, whether, what we are trying to do is that there are, in this chemical ecology project where we're sampling, the chemical signatures from both poop and from urine or spray, there are known chemicals to be associated with, specific diseases like, whatever, of course, cancer, whatever. And we're trying to see if we can see something like that, but we don't know. It's very exploratory.
Okay, and there's another question from Munish saying that by looking at testing the poop, can you say if the tiger is pregnant?
Yes. So, but not with DNA, but with these chemicals. So that's something we just, it's still very preliminary. We can't be sure, because that's why we're actually now, we started off by trying this out in Ranthambore, but then we realized that is stupid. We were going to do it anyway, but we're trying it out in the zoo where we know the truth. No, we know this individual is pregnant. We know this individual is a female. We know this individual is a male. And we have in the field also, we have detected chemicals associated with lactation, for example. So it is breakdowns of that. It's going to be very correlative, but we'll see how much we can get with that. There are also methods which we have not used, but to look at or to estimate age based on DNA. We've not tried them, but it is a possibility. Shall I go ahead?
Yeah, go ahead.
Yeah. So this is just to remind you that these are all the tiger reserves in India in black. Okay, these are the areas where tigers are protected, and roughly there's about 3,500 tigers in India. This is about 70% of the global population. But each tiger reserve has a mean or a median number, actually median, not mean, of between 40 and 50 individuals. So on its own, remember that figure we talked about extinction, 100 we said is some kind of a threshold. On its own, no population will survive, right? In the sense, it has a high probability of extinction. It's just a matter of time. And there are only 5 populations in all of these which have more than 150 individuals. So basically, we talk a lot about tigers. We see a lot of tiger pictures in the media. We hear people talk about tigers, but really there aren't that many tigers, right? And no population has lots of tigers, right? Think about the city of Bangalore, 11 million people. Okay, that's a lot of people, right? And there's like no population of tigers which has more than 150 tigers. That's kind of crazy, right? If you think about just the numbers. So like I said, Ullas Karant, and then subsequently the Wildlife Institute of India and the Government of India have counted tigers with camera traps. And we said, why don't we try to see if we can count tigers with their DNA just by walking through the forest, collecting these scats, bringing it back to the lab, and identifying whether they are different or same in terms of individuals. And this is one of the first projects I did in my lab. And basically it was in collaboration with Ullas, because the good thing was we worked in Bandipur Tiger Reserve in the Western Ghats, and the same time we were doing the genetic sampling, he was doing camera trapping. So there were two independent methods. One is genetics and the other is camera trapping, where we could compare our estimates to see, are they making any sense? Because we're not seeing any tigers, we're just collecting these cats. It's like kind of random, right? Like you just go collect these cats, you bring it back to the lab, you do some analysis, and poof, you come up with some number. Does that make any biological sense? Well, it turned out that in that year, and you can see here, for example, these are all the individuals and their recaptures. So you can see, for example, these blue diamonds, this blue diamond tiger was captured once and then recaptured twice. This blue, light blue circle was captured and recaptured once, and so on. So this history, this capture history, that allows us to estimate population size. And it turned out that that particular year, the number was identical between the genetic estimate and the camera trap estimate. So that was very comforting, that at least, there isn't some gross differences between the two approaches. So basically, like I said, tigers live in these landscapes. There are very few of these populations, but there is some evidence that they're moving between them. And some populations are isolated, whereas others are, large and potentially connected. So a lot of our work then focused on, because from a perspective of survival, these largish connected populations are the best. They have the lowest chance of extinction. But really, are they large and connected? This is something which we had to answer with data. So this is another way of looking at India. It's a Nightlight map from NASA. And it's kind of mind-blowing, right? To think about these animals, they have to move through all of this, right? To get from one place to the other. And you can see the cities. Whew, that's Nagpur, that's Delhi, okay, Mumbai. They're like lit up like anything, right? And like I said earlier, if you had very distinct populations, think about, say, the gene pool or the kinds of genes, they would be very different. But if tigers were moving, then these gene pools would be mixed. And so that's how we would analyze whether tigers are moving or not moving, right? And again, the same thing, walk through the forest. I have a fantastic set of people whom I've worked with. I've been very lucky, because a lot of these areas they're Naxal areas. They face many challenges when they're in the field, and I'm not there. They have to take spot decisions on their own. It's a lot of very adventurous, but also can be difficult. So you can see here, for example, a sample is being collected. The GPS location is recorded. This is for later. And, my student Prachi, who's of course now finished, is kind of explaining something to these volunteers. We work with lots of volunteers. All of our work is in the field. We have teams of 5 or 6 people walking, and it's always with volunteers. So if any of you are interested, you can always get in touch with us, later. And what we were able to show is that there is movement between these areas. And this is just to show that for these protected areas, we found signatures of, some kind of movement between them. And even more, what was even more interesting was that our data suggested that dense human settlements and high
Quick
traffic.
question, Uma, the earlier picture you showed of the movement, the
Yeah.
earlier slide, approximately what distance is this? I mean, are they moving like tens of kilometers, hundreds of kilometers, just so that people get an idea?
Hundreds of kilometers. This long distance potential movement, it may not be, it's not a direct movement in the sense that it's not like I identified the tiger here and then here. No, it's a genetic sharing indicative of movement, right? So that's why these dotted lines are not definitely animals moving, but definitely there is some movement potentially happening. And this is really far. This is on the order of 500 kilometers, right? So potentially it could also be like 3 or 4 generations ago. It's not necessarily tiger generations, which is maybe 20 years or so. So we can't say for sure, but what we can say is, what we can do then is to look at what is the habitat between areas where there is movement and when there is no movement, and ask what types of land use or habitats correlate with the presence of or the observation of potential movement or the lack of movement. Okay. And so when we do that, basically we find that there are two things which are bad for movement. Many things are not very bad. Agriculture is okay. Tigers move through it, right? What is bad for movement is basically high traffic roads and high density human settlements like urban areas. Okay. And so basically, if certain areas become barriers, if certain highways are built and so on, then what we could show is the chance of extinction goes up. So I'll show you one of them. It's the Kanha-Pench highway. And this is also, we also looked at effects of mines and so on. And we also suggested that the best way to keep extinction low is to have stepping stone populations. That is not just populations which are very far, but little places in between that tigers could move between. This is just a suggestion. It's kind of, again, abstract. How do you get that to work on the ground is a different thing. But in terms of minimizing extinction, that was the best way to do it in this landscape. And I don't want to, again, delve too much into graphs, but just to show you that if we look at a real landscape, because I said, conceptually, small populations go extinct, isolated populations go extinct, right? So in our context of central India, if we model extinction based on understanding connectivity from genetic data, what we find is that, yes, small populations which are isolated do go extinct. So this is, here you have population size, and here you have how isolated they are. Higher isolation index means they're more isolated, okay? On the other hand, largish and connected populations don't go extinct. So we said a threshold was 100, but even populations of the size 70 or so never go extinct. And these models or simulations were for 100 years. So in the next 100 years, we don't see extinction even of these largish populations, as long as they are connected. If they are connected and large, they don't. But there's a bunch of things in between. Many populations which could go extinct need not go extinct. And what happens to them in the next 100 years will depend on us, whether we allow them to grow or facilitate their connections to other populations. So this work then allows us to assess what is the best future for a specific. Some populations will always go extinct. It is a population called Tippeshwar. If you're from central India, you may have seen many fantastic pictures of this population and tigers here. The reality is it is very small and it is isolated. So it will keep getting recolonized. Tigers will come, but on its own, it cannot survive. It has to survive with some flow from elsewhere. But it will generally have a high extinction probability. It will keep going extinct. It will keep blinking on and off. So the other thing which this showed us is extinction is okay, right? It might happen. It might happen. You live in a short time window, but these processes operate over long windows of time. And so some things will go extinct, but for the whole system to sustain, there has to be this balance of connection and size. I'm hoping that wasn't too complex, but it allows us to really go from that very broad concept, like why should we believe stuff on butterflies in the context of tigers in Vidarbha, right? There's a very big difference we feel. But actually, these concepts hold, right? Irrespective of butterflies or tigers, Sweden or Vidarbha, they hold, and they also allow us to think about details of specific, populations. And this, I have discussed this with Arnab and many others before. This evidence allowed us, not us, but our colleagues from the Wildlife Conservation Trust. If you're in Bombay, you may know Anish Andheria, who heads the Wildlife Conservation Trust and has been pushing for RA milk colony and its preservation quite actively. But basically, with these colleagues who went to court, we were able to, through NGT, petition the Highway Authority of India to actually, we said that, if you're going to expand this highway, this is a picture from 2016, and I was driving on this road, and we had done this work, it was not yet published, that showed that when this highway becomes 6-lane, extinction probability goes up in that landscape by 50%, because this becomes a barrier. And on this side is Kanha, and this side is Pench, where Rudyard Kipling wrote Jungle Book. So basically, we were able to petition for this underpass, and the cars go over here, and then tigers can kind of go underneath, and all other wildlife. So the connection is hopefully maintained despite the highway. So this is what is called mitigation. That is, you can't not build the highway. People need it, but you can propose things which might not have as bad a effect. Okay, so I'm actually going very slow, so I'm going to skip a bit towards the end. But I said that these connections are important. Tigers have to move. The consequence of these connections is what? Tigers coming out of protected areas, and then you have like this cattle kill, and then the interface between humans and tigers increases, and this can lead to conflict, right? So this is my student Himanshu, is a very brave fellow. He's been working outside of Bandhavgarh, and this is a picture from outside Bandhavgarh, where he has actually now collected samples from 400 cattle kills, because there are many questions. Are all tigers killing cattle? Are some of them coming outside and killing cattle? Is it nothing to do with the tigers? Is it to do with some areas having lots of cattle and every tiger will come and kill cattle there? Again, how do we answer these questions? We can't follow them all the time, right? So if we sample the cattle kills and try to understand who killed that cattle, and we do this over many cattle kills over a long period of time, we can try to infer what correlates or what are important things which go together with a lot of cattle kills. So this is a really nice picture from a camera trap, but you can see the tiger here. And can you see actually what it's doing? I don't know if you guys have cats, but I'm sure you've noticed with your cats that when the cats have their prey, one of the things they do is they lick it. And this tiger is actually licking the prey. And so that's good for us, because when it licks the prey, what does it leave on the prey? It leaves its saliva. And saliva, like poop, is also a source of tiger DNA. Okay, so what we can do, and I'll just show you this video. It's pretty cool. You can see this tiger is here. This cow is, of course, dead, and it's really licking. It's really looking. So they are very, cats clean their prey also before eating it, and so basically what we can do is we can find these bite marks, lick marks, and you can see here the skin is very flattened here of the cow. That's where the tiger has licked, or leopard or whatever, we don't know what it is, right, has licked it. And we can find lots of other stuff around this crime scene in some sense, where the predator has actually killed the cattle. There are lick marks, bite marks, there are fecal samples. And when we come to this kill, we can actually come and collect all of this material, take it back to the lab and identify, are there many tigers killing cattle? Is it the same individual? Or, how does this change over time? So this allows us to put in some kind of a framework, right? To say, okay, we were able to identify the same individual in multiple places and so on and so forth. So I guess, Arnab, what's the time?
Right now it's about 11:54. Okay. I don't know if you want to take a break. There are lots and lots and lots of questions.
Okay, I'll go ahead and take them.
So maybe because we were dealing with isolated populations, etc. Yeah. So on on YouTube. So, how do you define an isolated population? Is it like how many tigers there are per kilometer square or something like that? Or, in that region, how do you define isolated as one? Related to it, Paritosh is asking, Amit is also asking, do tigers actually like to be lonely and isolated? And does it also depend on the mental state? I mean, maybe a tiger just wants to be isolated. Is there anything like that?
Yeah. So, it's a very good question about how you define isolated population. And we struggle with it all the time because scientifically, one of the ways is, like you said, like, for example, I won't be able to speak about it today, but this study, which we are going to have come out soon, and I hope I'll get a chance to speak about it later, on Simlipal Tiger Reserve. We thought Simlipal was isolated, but how do we prove it? We don't know, right? So basically what we did is we went to all the areas, any areas where they were thought to be tigers, right, around. And we sampled there and we looked at whether there were tigers or not. And all of that showed us that there were no tigers. We can use genetic methods. So for example, if we take genetic samples from all of these areas and also from here, and we put all the data together and we see, does everything stick together? Does it look like a nice continuum, or does something stick out as different, right? Has a different genetic signature or profile. That is one way to tell. Because if it was not isolated, if there was gene flow or movement, genetic mixing, then everything would look the same, right? But if something looks very different, that means there has not been mixing. Okay. And then finally, we can do these connectivity analyses where we say, okay, this population, let's look at the potential flow of individuals from all these other populations, given the habitats and what we think is conducive or good for tiger movement or not, right? So these are various ways we can do it, but you're right, we cannot really prove that, unless we watch all the tigers and make sure they don't move or something like that, right, for the next 50 years. So the time scales of these things are long, and so it might be a little challenging. Yeah. And so for example,
there's one more question. What is the meaning of isolated? So isolated generally means there is no easy contact between this group of tigers and a group of tigers at a somewhere a little further away from them.
Yeah. And the thing is, the word isolated sounds how should I say, very final, but you can see it's a continuum, right? For example, if you look at this population, Ranthambore, you would be surprised, but it is isolated from, this is Sariska, and between Sariska and Ranthambore is the town of Alwar. Big town, lots of people, lots of highways. Between Ranthambore and this population is the Chambal, and the Chambal is a valley and a river, and some tigers have crossed, but statistically over time, there hasn't been a successful movement which resulted in mixing. So the word sounds simple, but to actually prove it is very tough. And so we are just kind of using a moving target, kind of, okay, to the best of our understanding, to the best of the data we have, it looks like there is no contact between this and some other populations. Okay,
so that is probably...
and there was a question about being lonely. No, tigers are solitary. Yeah. They are solitary. We know very little about tiger behavior. Like when we say, like when we look at our cats or dogs, we know they have personalities, right? I don't think we know that much about tigers, that they have personalities. We can anthropomorphize, we can, sort of think we know, like there are some tigers, famous ones like Machli. She was supposed to be very aggressive. She was had a very prime territory in Ranthambore, and a lot of tourists saw her. She's the most photographed tiger, tigress. But the thing is, you saw her and you said she was aggressive, but what about the others? You've not sampled them. You don't know about them. So I'm not sure, to make a statement like that based on scientific data, would be tough. You have to first have many tigers whom you look at, and you have to also be able to score. What do you mean by like to be lonely? How do you scientifically explain or quantify like to be lonely, right? Does that mean that individual is never with other individuals or something like that? So as a scientist, we have to think about how do we answer these questions with data. I'm not dismissing the question. I'm just saying that it's very hard to answer some of these questions. Is that okay, Arnav?
Yeah, that's good. And there are 3 questions which are related about, you showed cattle kills. So can you determine, Munish is asking, can you determine if the kill is done by a tiger or other predator from claw marks or something else? That was on YouTube. And I think on Zoom, there was a similar thing from Sana, who's asking, how do we know who killed the tiger or who killed the cattle? Is it something related to enzymes on the teeth or saliva? I guess it's who killed the cattle, I guess.
So that's what we're trying to figure out, right? See, what happens is that most of the time, what does the forest department do? So the thing is, as soon as a cattle is killed, the villagers or the owners of that cattle file for compensation. Okay. And they get a compensation for every cattle because naturally, they should get. But the thing is, what the forest department does is they put up a camera trap. And by the way, have you guys watched Sherni? You should watch it. It's a really fun movie. And some of this comes out in that film. They put a camera trap to try to see who is the individual. But when we were observing a lot of camera trap data from these kills, we realized that many times after the kill is made, if it is close to the village, the same tiger may or may not come back. Whether the same tiger comes back or not, many individuals have been known to come back to that kill. Okay. So the camera trap data doesn't, and sometimes the kill is abandoned also. It's too close to human habitation. There's too much of disturbance and the animal doesn't come back, right? So from this camera trap, we cannot be sure who the true killer is. Okay. The true killer of that cattle. Now, the best way to be sure of the true killer is to be able to get genetic material from the jugular bite mark. The jugular bite mark, attack on the jugular is how carnivores kill. Okay. So basically, and it's a very ingrained thing. I have a cat and it always attacks the stuffed toys also at the jugular. So basically, if you are able to, and those bite marks, amazing. This student Himanshu, he's like really creative guy, and he actually said, no, Ma. I said, come on, don't be crazy. We're not going to get DNA from those bite marks. That just doesn't make sense. But he did. And we are able to, not always, but from those bite marks, we can get DNA to identify individuals. Okay, so that's something which is just not yet published, but I think it's something which is pretty interesting. And if we can understand something about conflict, it's very important, because I can sit in Bangalore in my AC office and say, we must support connectivity, but I'm not the one whose cattle is getting killed every day, day in and day out, right? So as these animals move, there's a lot of conflict and we need to understand it better. I hope that's...
So when you say you can identify an individual, it means that the DNA from this place, the bite on the neck, is the same as the DNA I have collected from a poop sample sometime earlier. So this tiger is in my DNA database. But
exactly.
It still does not correlate with a picture of that tiger.
Exactly.
That this particular tiger. You know that this tiger is one of the tigers in this place, but that's about it, right?
Arnav, you caught me. Yeah. So you know what? That's why we're doing this. Sorry, let me go back. You see this thing here? That is a piece of cardboard with double-sided sticky tape. And when the tiger came to eat this cattle, it stepped on it. Okay. So we not only got, we got its DNA, that particular tiger's DNA. And the biggest challenge is going to be linking that genetic database to real life animals, right? Like, who is that tiger, right? I mean, like, if you have my DNA, you don't know it's Uma Ramakrishnan, it's some You can say, oh, South Asian of, South Indian ancestry. You can say a lot of things about me from my DNA, but you may not be able to match it to me as in me, right? So these kinds of databases are becoming more popular globally. I'm talking about even with humans. The UK, for example, has a policy to collect DNA from anyone at any time. In humans, there are a lot of ethical issues associated with this, and people have been, for example, talking a lot about the DNA bill in India, which also is aiming for this, and it's been passed. But I guess we would have to have some way to get with a camera trap image genetic material. So one way is the sticky tape kind of thing. Yeah.
But the sticky tape, the paw prints are unique, or
no, paw prints are not unique, Arnab. That is what for a long time people were trying to count tigers with paw prints, and they're not unique, but the hair gets pulled off. And so we can get some hair, and it's been working pretty well. So of course, my student Himanshu is like a diviner. So he basically like looks around. He's like, okay, the tiger's going to come from here. So let's put some tape here. And it's like, I just watch because it's quite fun to, you're kind of thinking like what might happen in the night, because you're not there at night when all of this action happens.
Okay, so I think the other questions are all general questions. I think we'll come to them at the end. I don't see anything more about.
I'll just show one last thing. Yeah, if that's okay.
There's still lots of questions. Don't worry.
I won't show this stuff, but we've done some work on Ranthambore where we're looking at inbreeding. I can talk about it another day. We've done some work on these beautiful pseudomelanistic tigers in Simlipal. That's why we're working on them to look at what is the genetic basis and so on. And I'll talk about that also some other time. Why does this tiger look this way? And in Ranthambore, we're trying to reconstruct the pedigree. And this time we're actually doing it with hair, just because we can see a tiger, and then when it sits down and moves away, we find lots of hair. So I won't discuss this in detail, but I'll give you this. I'll just show you this a very funny story. Sorry, there's something wrong with the animation. Sorry. So this little cub, here, this cub, there was a teenager I think it was a couple of years ago now, drove across the US-Mexico border with this cub in the passenger seat. Okay. And like totally illegal, right? You can't take an endangered species across the border without any kind of permit. So the cub was confiscated from the teenager. And the first question was, it was unwell, it was dehydrated. So it had to be taken to the zoo. San Diego Zoo is right there. So they took it to the San Diego Zoo, and the zoo people were taking care of it. And then the question was, what do we do with this cub? Because in the US zoo system, the US zoos, the American Zoo Association has decided that they are only going to breed 3 types of tigers. That is Amur tigers from Russian Far East, Malayan tigers from Southeast Asia, and Sumatran tigers from Indonesia. They don't keep Indian tigers because they feel there are many tigers in India. The other tigers, there are much fewer in the wild. So they are focusing on the genetic conservation of those others. So the question was, who is this cub? Like, can he, it's a male, go into some zoo because he is Russian tiger or whatever. Or what do we do with this tiger? So the funny thing was, because of our work on tigers, and since I know people at the San Diego Zoo, they contacted me and said, can you help us figure out the ancestry of this tiger? Okay. So they were able to collect some blood samples. In this case, we actually sequenced the whole genome of this little fellow in the US, right? And because we had genetic data and genomes from tigers from India, from Indonesia, from Malaysia and the Russian Far East, we were able to compare and ask, what is this tiger, right? So you can see here, for example, this green are one group, right, which are basically tigers from the Russian Far East. This blue are Indian tigers. They're all more similar to each other than they are to the Russian Far East tigers. This red is the Malayan tigers or the Southeast Asian ones. And this mustard color is basically the Indonesian. And if you look at this cub, okay, this cub was a nice mixture of everything. So this cub then, his fate was that he had to go to a sanctuary or a rescue center, like a goshala, like a home, okay, which they have here in the US. They don't have them here, right? They have some rescue centers, but very few. So basically, because he was not a purebred tiger, he went there. And the reality is there are more tigers in farms on Texas, farms in Vietnam and China and so on and so forth than they are in the wild. Okay. So who are these tigers? What is their ancestry? We know nothing about all of that. So I told you a lot of conservation stories of tigers in the wild, but there are a lot of tigers in captivity as well as in these illegalish establishments, and we don't know a lot about them either. But this was just a fun story. We got to interact with the US Fish and Wildlife and San Diego Zoo, and genetics was used for something completely, different, which decided the fate of this poor little guy. So I will also, just because there were some questions about how can you do, what can you do, and so on. Me and several others were part of the Biodiversity Collaborative, and we are trying to propose a national mission on biodiversity and human well-being so that India as a country can really mainstream the importance of biodiversity. And I hope that if it comes through, there'll be a lot of opportunities for public and others to participate in a lot of these studies. So I'll stop there, and if we have time later, I can show this book. We cannot, the work we do is of course made possible by lots of money.
So, Uma, if you want to show the book, you should stop screen sharing. Otherwise, your window is a very tiny window. Remember that.
Okay. I'll show it in a minute.
If you want to really show, so
I
you can acknowledge people and
out,
then maybe show the book later. Yeah.
I just want to say that, this guy here is Mujahid, and he's been working with us, and this book is by him. I'll show you the book. So he is from Savai Madhopur outside Ranthambore, and he's been working on tigers over 10 years with many different research teams. So his knowledge of tigers and his stories of tigers are mind-blowing. And so a lot of the work we do, we publish it or, talk about it, and I'm talking about it, but it's done by a lot of other people, and, they don't necessarily always get acknowledged. So I just wanted to mention there are many, many people who contribute to the work we do. So I will stop screen sharing now and just show you that what we did with Mujahid was to actually, he came down to Bangalore and he met this team from Pratham, and they worked with him to write a book. So he's actually it's a little difficult with this blurring. He's the author of this book.
Below you, yeah.
Yeah. And so anyway, it's called Tiger, Where Are You? It's a reader level 1 book, not something which, has come from the research, but from the journey and the experience that we've had. So thanks for listening. And I know I went very over time. So happy to.
No, not at all. It's been a pleasure to listen to you. Let me look at some questions right now. So you talked about this person who brought a tiger along with in the car or something. So at the end of it, Sham is asking, how did they figure out which country the tiger belonged to? I think the answer was it was mixed, right? It was not a particular...
Yeah, so the thing is, basically, there are a lot of people who breed tigers. There are suggestions in Mexico because tigers are a symbol of power, right? So for example, these magicians, Siegfried and Roy, they're very famous magicians from Las Vegas, and they had a major tiger breeding operation because they used to have a white tiger as part of their magic show. So there are a lot of people like that who breed tigers, and this is probably one of those kind of bred tigers.
Okay, so since you talked about a white tiger, right from the beginning in the question answers, there have been a lot of people who have asked about, let's see the questions. I think they were in the chat here. There are questions, about essentially how do you, so Vivek has asked this question. I think earlier somebody else had asked it as well. How do you have these white or black kind of tigers, not the standard orange and striped ones? How do we get these different colorations in tigers?
Basically, they're mutations, spelling mistakes in the DNA, which result in these, phenotypes or looks. And the work we have done for the melanistic or the black tiger, it's not completely black. So it's pseudo melanistic. It's not really melanistic. Is to find the mutation. And similarly for the white tiger, the mutation was actually found by a team from China using white tigers from the US. So you can find these mutations by looking at pedigrees, and identifying them. And then you can, of course, so basically that's how you do it. Yeah.
Okay. Let me go back. So by the way, people who are monitoring Facebook, please interrupt and let us know if there are questions from there. Okay, so let me go back to YouTube where I think there's a whole bunch of questions. Can the age of a tiger be identified from the scat or the hair?
So I said that
Jyotika asking that.
Yeah, so there are some new ways based on centromere length for those of you who are biology students, but we haven't tried them. They've been used in many wildlife species, but we haven't tried them. But it's possible, it's unlikely to be able to do it very well from scat because the DNA in scat is degraded, or it's kind of broken up. More likely probably from shed hair, but we haven't tried it, so I don't know.
Okay. Priyatosh wants to know, do these tigers in different places have different preferences for a particular prey?
Yeah, so that's
do you have data for that?
Yeah. So we don't have data for that yet. We would need to do diet studies across the country. We do know, for example, that in the Sundarbans, tigers do tend to eat more fish and drink brackish water sometimes. But I think that is something which would require a lot of detailed investigation over time, because diets can also change seasonally depending on what prey is available and so on. But in general, tigers like big prey, and if present, their favorite prey is gaur or the Indian bison. However, the gaur is not present in all parts of India, and should the gaur not be present, they will take smaller prey like sambar and so on. But they do also eat even smaller prey. So they are kind of quite general. It's not like they won't eat cattle, or they eat cattle also. So it's not like it's a predator. So they'll eat whatever is there, but they're not as flexible as say leopards, who have been known to even eat sometimes insects. And leopards actually probably subsist quite a bit on dogs, feral dogs, of which also there are lots in India.
Okay, Aishwarya will come to your question. Let me take the ones which have already been typed in. There is a question from Daman Bha. Oh, I think Daman is from Shillong actually. Says, endoparasites can cause severe harm to the health of a tiger. Have you ever encountered parasite eggs in tiger poop?
Yeah, there are lots, and we would love to study this, but we've not done it systematically. And we would like to understand, for example, in the population we're studying in Rajasthan, Ranthambore, where we see inbreeding. One prediction is that inbred individuals are less fit, and one manifestation of this might be higher parasite load, but we have not studied it yet. But that would be very interesting to investigate. Thanks okay. For that.
Yeah, Shubham and Priya, common question. Do tigers have rabies virus, and do tigers also host other viruses which can be bad for them?
So this is again something which we don't know much about. Yes.
And if they kill a rabid dog, can it be transferred to the tiger? Okay, so 3 of them on rabies.
Yeah, there is no reason why not, but we haven't seen cases of rabies in tigers yet. Okay. And yeah, so I guess that's the right kind of answer. But some other known canine viruses like canine distemper have caused outbreaks in cats in general, in lions in the Serengeti, but also in tigers in the Russian Far East with high mortality. We do not know of the current health risks in terms of viruses for tigers in the Indian subcontinent. You may have noticed recently there was a directive to shut down all tiger reserves to tourists because of the death of a lion in in Chennai, in Vandalur Zoo, from coronavirus. So usually the thought is that uh, cats don't die of coronavirus, but they do get infected with it. So I think we just have very little knowledge of these things. The thing about studying viruses is that many of these, we're talking about coronavirus, rabies, canine distemper, are RNA based viruses. And RNA in general is very finicky, unlike DNA. It degrades very fast. So there are technical challenges then with studying these kind of viruses, but it should be done. No one has done it yet.
Okay. Harshita is, okay, this is probably just a fact. Is there a tiger named Munna in Kanha National Park with some marking on his head that looks like cat?
I mean, that's what, so basically people really love tigers, and so they name them, and they look at these distinctive features. And as a scientist who works on populations and genetic variation of populations, we are not really looking at specific individuals like that. We look at trends in populations. In the case of Ranthambore, where we're trying to reconstruct the pedigree, our exploration is more individual based, and it is coming to that in this conflict study. Yeah, so usually when we approach these questions, we don't think of them as individuals. So that's how public usually sees tigers.
Okay. Okay. Ooh, that's a very long. Aishwarya Kashyap, do you want to unmute and ask the question? I mean, this is like a thing. Okay, I can try and read it out. Let me take a very quick one first. Do tigers scavenge?
Sometimes has been seen, but not usually. Not usually. I mean, they're definitely not like hyenas, which hyenas will eat tires. They'll eat anything, right? So they're definitely not like that. But I think there have been some reports of scavenging. I can read out Aishwarya's question. Basically,
yeah, I've asked Aishwarya, if you want, you can unmute and ask. I have allowed you to talk. Yes, Aishwarya. Yeah, go ahead.
Go ahead, Aishwarya.
Hello, ma'am. Good afternoon. Actually, I have a question that, I know that tigers are are endangered animals, and we need to protect them. But my main question is that, since the human population is increasing day by day, and we are just cutting down trees, and we are the habitat is getting lost for many wild animals, what is the solution for that? As an individual, what can we do? And the sub question is that not only the habitat loss, and we are ill-treating animals, poaching them. Some people are poaching them, and mainly except from that, there is also air pollution, water pollution, and the animals are also getting affected by that. So what, as an individual, what can we do, ma'am?
Yes, that's a tough question. As an individual, what can we do? I can think about it. I can probably answer it initially from a perspective of a strategy for the future. So basically, this year is the year of restoration. We have a lot of habitat land in India which is degraded. It has been cut and left, and so on and so forth. And because so many people depend on nature for their livelihoods, you take whole of central India and mahua, a lot of people go into forests to collect mahua, right? And that is their livelihood. That is how they live. And there's a lot of people, we just ignore them because we live in cities, who depend on nature for their incomes. So if we are able to do restoration, I'm not saying everything should become a tiger reserve. Obviously, that's not possible. When I say tiger reserve, that's like a highly protected for tiger only type. It's not like that, but space. It's not really like that. But so I think that some kind of vision for the future, which is not, I'm not saying anti-development, but development in the context of restoration and some kind of a balance with nature, I think there is no other option. Now, whether there is a will to do that is not in our hands. And sometimes I can say from personal experience, I sometimes think, what is the point of doing any of this if nothing is going to work? But there's no other option. You have to do what you can do. And what happens, it may not be in your hands. So as individuals also, we have to make sustainable choices, not simple things like how we use plastic, what we do. Those things also add up and affect the entire cycle of climate and so on. And we have to try to do what we can as individuals. And maybe this is a bit forward of me, but to really raise our voices to say that this is what the future should be. We want a sustainable future, because many times politicians parrot that we only want development. And that may not be the case, actually. The people, not everybody is consulted in these things. So I think if we really believe, it depends on what you think. I can't tell you what to think. But if we feel that it's important to have a sustainable future with a balance with nature, we need to say it so that people take it seriously. Enough people need to say it. I don't know whether that was too philosophical an answer, but this is probably a philosophical point.
Okay, thank you, ma'am, so much. Thank you.
Yeah, I think Dharmendra also had a very similar question, given the balance between social economic development, environmental issues, and conservation. What happens if we, do you think the carrying capacity of the tiger reserves will be reached very soon, and what happens after that?
Yeah. I think that tiger reserves, some of them, I'm not an expert in tiger ecology, but some of them are at carrying capacity. Several reserves in Madhya Pradesh and Maharashtra. But on the other hand, areas in Jharkhand, Chhattisgarh, West Bengal, Odisha, there's much potential for protection, increase in prey, and tiger numbers. There are protected areas which are devoid of prey because of the lack of socioeconomic opportunities, potentially, which results in more hunting and things like that. So there has to be some effort to ensure, prey protection, persistence of tigers with human socioeconomic development, so that, without that, there's no option at all. So our model has to be of coexistence. We cannot prioritize humans or tigers or this or that. This model of coexistence is something, it's new, and it's something which India can take a lead, because overall people in India are very sympathetic to nature and wildlife compared to most parts of the world. In a very so-called, high seat of excellence like California, Palo Alto, if there happens to be a mountain lion which comes in, it's immediately shot. Okay, that does not happen in India. There's a lot of tolerance. Our people have a lot of tolerance, and so we have to work to kind of build this somehow in a way which is quantitatively beneficial to them also.
Okay, so I think I'll take two more questions. There's one from Priyatosh, who says, when you interact with villages and villagers, and they see a tiger, what, are there some sort of, do you tell them what they should do or something? Are there some better ways to sort of interact and not be like people in Palo Alto would shoot that thing immediately?
I'm sorry, I demonize them, but I guess, I'm not at that, I'm not a person who's boots on the ground, right? Like there are people who are grassroots who work on the ground. We are scientists, we go in, we work, we leave. So we don't have those long-term relationships. Maybe sometimes we will develop, like in Ranthambore, we're trying to develop. I don't know, I would love to explore how that can grow, because as a scientist, I don't actually know much about many of these things also, in terms of, giving advice to someone. I mean, in the sense, it's their life and I can't say, no, save the tiger. I mean, it seems a bit forward of me probably to do that, to tell them how to live their lives. So I feel like maybe, interdisciplinary teams of scientists should work, interdisciplinary teams of researchers and multiple stakeholders should work on these things together. And there should be a very strong local presence and engagement. So I didn't really answer your question, but I think it's something which, I think if that's a very basic question, like what should you do when you see a tiger? Well, of course, just try and make sure the tiger doesn't see you. And usually cats and things, they just go away. For example, lots of times in Kaziranga, there are floods. Every year there are floods, it's that's the way the system is. It's a flooding system. And tigers, come into people's houses and people just move out of the house. And after some time, the tiger goes. So you can, if you leave them alone, they'll go. But if you create a lot of hangama, a lot of people watching it, taking selfie, those situations make animals scared and then they also attack. So those are things which we've seen. You might have seen a lot of images recently of people throwing firecrackers at elephants. It's crazy, when you have a lot of agitation of animals like that, then naturally they will lash back. And then you say, oh my God, this animal is lashing back. So I think those are things which we need to sensitize more. But sometimes there's also a lot of anger against animals. Because, for example, in places like Pilibhit, where there are tigers that live in sugarcane fields, and a lot of people get killed by tigers. There's a lot of anger against animals as well there. So that's why if we study conflict, if we can minimize those bad situations, at least that would be good. Because once the people turn against the animals, that's it. It's very hard for them to survive.
Okay, so I think we'll take the last questions from Sana, who's asked many questions during the thing. One is purely technical, which should be pretty quick. And then I think she has a very nice closing question. So I'll end with that. The cub which came through the US-Mexico border, that same back to that, it was not purebred. Hence, does it, because it has more genetic diversity, is it more likely to have a chance of survival?
That's a great question. And that's something which we are not addressing yet. Because if you really say that genetic variation is money in the bank, then these individuals, they're basically outside the system of conservation. That's why I kind of also brought it up in the end, that there are all these individuals, we know nothing about them. Maybe there's a potential for rescue or infusion of genetic variation with these animals in the future. They will never contribute to tigers in the wild, they're not going to be released into the wild. That's what I mean. But maybe their variation is something which we can think about characterizing and thinking about how it can contribute to tiger conservation in the wild.
Okay, and Sana, you also asked that, you said a lot of the work is done by volunteers. So I think a good last question is, how do people volunteer for such projects? And, how does that happen?
Yeah. So, I mean, you can write to me. And the thing is, of course, I should give a caveat that we've not been able to do much field work for the last year or so, year and a half. But hopefully at some point, we are also not at this point specifically taking on lots of volunteers, because I don't want to endanger the lives of people. Sometimes in these smaller areas, we may not have very good medical facilities and stuff. So, but I think that you can write to me and I can always, forward it to my students. But I think maybe we should also set up a more formal way on the website or something where people can just write in. But yeah, so.
Okay, so with that, I think, we are past 12:30. Thank you for taking all these questions.
I can put
your
my
email. I can. We can put it in the chat. Okay, but there have been many comments saying thank you, both on Zoom and on YouTube. So thanks a lot. This was extremely not just informative, but thought-provoking. And how does one actually deal with something like this, where humans and animals are involved? Clearly, there are no easy solutions, and it's interesting to see how science can help. Find, at least get the data based on which we could make evidence-based decisions. We can help influence policy based on scientific evidence. I think that's very important. Thank you, Uma, for being with us. For everyone else, just a reminder that August has 5 Sundays. So we will have a special 5th Sunday Chai and Why 2 weeks from now. So we'll announce the topic on Facebook, Twitter, whatever. Follow us and send us an email if you'd like to be in our mailing list, and do spread the word. Sundays, 11 o'clock, there's Chai and Why on the 1st, 3rd, and 5th Sundays. Till then, stay safe. See you. The pandemic's not over, so still, please stay safe. Get vaccinated if you can. Cover your mouth and nose when you go outside. And see you later. Thanks for watching. Bye-bye.
Thank you. Thanks, Arnab. Thanks, everyone. Bye.
Bye.